All right, let's go ahead and get us rolling here. Last human anatomy lecture of the semester. We made it almost. We got today's lecture, and then you have your lecture exam Friday and over the weekend, as you like. And then we have our lab practical on Tuesday. And then you never have to think about human anatomy again. Just kidding. So our last class, our agenda looks like this. We're wrapping things up by being unexciting and predictable, which means we'll have a review drawing activity, talk about the respiratory system. I have a few videos for you. Then I want to debrief your quiz. I have those graded. I'll give those back to you today. And then just some schedule reminders. I know that it's not Friday, but since it is our last lecture, I do have a Venn diagram on your way out. So get us thinking about the respiratory system again. Here is a diagram. Go ahead and I left most of the stuff labeled because we haven't talked about it yet. But go ahead and draw this diagram and fill in 1, 2, 3, 4, 5 blanks. While you're doing that, I'm going to get your quizzes organized. Yeah, some of these blanks are getting a little bit ahead of us, but that's okay. You might remember them from lab, and if you don't know it, then it'll be a nice introduction anyway. Hey, Julian. All right, a couple seconds, get down what you can. I see a lot of you still looking and drawing, so if you need a couple minutes or a couple seconds after the timer goes off, that's okay. All right, let's have a look at what those missing structures are. So on the top left there, you see your nasal cavity. Remember, that's this big area here, superior to the palate, and you can see the palate there. You might remember this from your cats. Notice a big difference is that in the cats, we don't have this 90-degree angle here. It's essentially just a straight line because they're quadrupedal, and so everything is structured in a line. It doesn't have to bend like it does us humans. So note that in the cat, this nasal cavity and the nasopharynx are kind of one big continuous structure. Bless you. We don't have as much of a division as we do. And so then notice here we have the nasal cavity up here, superior to the palate. And then after we get around this bend, we have this area called the pharynx, which is where the nasal cavity first flows down towards the respiratory system. Oops. And then what were the other blanks? Then we have the larynx here, also known as which includes, I should say, the voice box. So this is the area just past the epiglottis and the glottis, where the digestive tract and the respiratory tract overlap. So remember, the epiglottis is the flap that closes over the glottis, which is the hole. Super important for not getting food and drink down your breathing tube. Let's see, what else was missing from the diagram? Oh, and then of course we have the trachea. And that should be readily familiar to all of you. That, of course, is also known as the windpipe that connects the upper respiratory airways here down into the lungs. Remember, the trachea has these cartilaginous rings that give it structure, so it stays open even as pressure changes. So think of it like the tube for your vacuum cleaner. It has those rigid rings so that when the vacuum cleaner turns on and sucks, it doesn't just collapse the tube on itself. That's just a start orientation to those major structures. We're not going to get into too much detail with them, but I do want you to be able to orient yourself to those large structures. And we'll talk a little bit about the alveoli today at a smaller scale. What questions do you have before we kind of jump in? Cool. All right, so we can remember this is review. We can divide the organs of the respiratory system into the conducting zone. Remember, the conducting zone, no gas exchange happens. Instead, that air is subjected to 3 processes. It's filtered, it's humidified, and it's warmed. Then we get into the respiratory zone, and that is where gas exchange actually happens. So let's back up. Last time we left off, we were talking about the conduction zone, and we talked about the nasal cavity a little bit. We just started to get into the pharynx. So let's pick up where we left there. And yeah, the pharynx. So the pharynx is this funnel-shaped passageway that connects the nasal cavity and the mouth. So it's basically this spot right here. Important to note that this is only going to contain air. The opening for your mouth where food and water pass down to the esophagus is down here. And so ideally, anyway, the pharynx is only going to contain air. Of course, under some extreme circumstances, you might get food and water up in there. And then you can divide the pharynx into 3 sections. We have the nasopharynx. So actually, let me clarify. I got a little ahead of myself. The nasopharynx, the superior area here of the pharynx, contains just air. So let me clarify that. The pharynx is all of this structure here. The nasopharynx is that top part where we have just air. The oropharynx is where food and water can come in. So we get both mixed there. And then we have the larynx down below. And this contains air and food. But this is where we have that transition with the glottis and the epiglottis. So the food will go down the esophagus and the air will go here. So we have this area here between the oropharynx and the laryngopharynx where we have food and water mixed. Although generally they're not going to be in there at the same time. We don't. We try not to breathe when we eat and eat when we breathe, vice versa. Of course, we're kind of alternating those things in rapid succession because you can't hold your breath for too long. But generally, your oropharynx is on one duty or another. So pharynx is all 3 of those pieces. It's divided into these 3 sections: nasopharynx, oropharynx, and laryngopharynx. In our cat, the nasal cavity and the nasopharynx were kind of continuous because we don't have this bend here. So it's kind of all one thing, more or less. Because of the different substances that are passing through these areas, the mucosal lining differs along the length here. I'm not going to get into the detail, but I want to put that up. I want you to remember that all of the lining of the pharynx is lined with these thick mucosal lining that again serves a function to clean the air as it passes down, to pull out particles, bacteria, etc. And then what ends up happening is you end up swallowing that down in your esophagus. So you're constantly producing mucus. That mucus is constantly grabbing the gunk out of the air, and then you're constantly swallowing and digesting all of that gunk. Awesome, right? So let's talk a little bit. That's actually all I want to say about the pharynx. The next section of the conduction zone is the voice box, or what we call the larynx. So this is just distal or after the glottis and epiglottis here. And we can see what the larynx looks like from a anterior view. Notice the, actually, let me just put those up. So it extends from about the 4th to the 6th cervical vertebrae. So it's kind of right in the center of what we commonly call your throat. And it attaches to this hyoid bone. Now, we talked about the hyoid bone a little bit in our skull lab, but very, very briefly. But this is kind of a free-floating bone. It doesn't directly articulate with any other bones, but rather serves as an important attachment site for muscles associated with swallowing and speaking and moving your neck, and also provides some structure for the larynx. a little bit of hard structure for the excuse me, not the larynx. Yeah, the larynx. The larynx then continues inferiorly and transitions and becomes the trachea there. In your textbook, you'll see specific names. This is something called the cricoid cartilage, and you'll see specific names for these cartilaginous pieces. I'm not going to get into that. I just want you to know the larynx is the voice box. It's the transition between the pharynx and your trachea, and it's associated with the hyoid bone. The larynx is lined with epithelial tissue that is stratified squamous cells superiorly, but transitions to pseudostratified columnar cells that are ciliated inferiorly. So up top, there's a lot of friction as air is coming in and out. So you have these squamous cells that are stratified, so you can lose them and be replaced by cells underneath. But as you go down and then into the trachea, you get these pseudostratified columnar cells. And you saw this in lab yesterday. The first microscope station had slides of the cross-section of the trachea. You could see those pseudostratified cells really well. You can see their little cilia if you look closely. The larynx has three major functions I want to cover. One is it's commonly called the voice box. So it is where many of our sounds are produced. Without getting into it, it's important to note that our speech is produced by a combination of sounds from our voice box, but also the way we move our mouth and our tongue and our teeth. So for example, if you think about making the letter S, a lot of that sound is the front of the mouth where the tongue is coming into contact with the teeth. Whereas if you're making like a vowel sound, like an oo, that's coming from your voice box here. And it's a combination of sound production from all these different places that ends up being speech. The larynx also provides this cartilaginous structure, provides an open airway for air to pass through between the pharynx and the trachea. And as I mentioned, the superior most aspect of the larynx contains this epiglottis that is a flap that makes sure that food and water don't go down the air tube. And so let's look at that in a little more detail here. Actually, I'm going to skip over a little bit of this. This is just about the vocal folds that make your sound of your speech. I'm going to skip this slide for now. I'm just debating how much of this I want to cover given our time today. Sorry, all right, I'm going to actually stop there with the larynx. I'm not going to get into the details of the vocal folds and how sound production is made. We'll save that for a future class. I do want to note one thing though, and I'm going to skip ahead here. Don't worry about the details. What I want to just bring your attention to is that the larynx can actually close not only to prevent food and water from going down, but also so that you can create pressure in your lungs and the air doesn't escape. And this is something known as the Valsalva's maneuver. Sometimes we refer to it as bearing down. This is what you do if you're when you're close off your lungs, but you kind of contract your lungs at the same time and you build up pressure. And so, for example, if you're trying to lift something heavy and you're like doing that thing, that is called Valsalva's maneuver. It has important biological functions as well. It can increase pressure in your thoracic and abdominal cavity. For example, if you're straining to defecate, we've probably all been in that situation at some point where we're using this. It can actually change the pressure in your thoracic cavity to regulate your heartbeat a little bit too in an interesting way. So I do want you to know that the larynx can provide a seal for air from escaping so that you can create pressure in that cavity that can have some important biological functions. Go ahead, Jada, I think your hand was up.
Oh, I was just curious if that's like what's happening when you have like a panic attack. Like that's like that beating in your chest, like you feel like right before you have a panic attack.
I don't know what the physiology of that is. My guess is probably a combination of things. It's probably increased heart rate, and then in some cases it can be like restricted breathing. It might not be from this part of your airway being constricted specifically, but it could be. Yeah, that sounds pretty reasonable, I think. And it's probably different for different people too. Yeah. Good. Yeah, go ahead, Julie.
Yeah, that happened to me. I used to dance,
Mhm.
and then it happened one time that I went to the doctor after, and they told me that was it. My throat was so dry that the epiglottis couldn't like open again, so it closed and I couldn't breathe.
Oh my gosh. And so what did you, how did you fix that? Or what happened?
I drank water.
Okay,
Literally, I couldn't breathe, so they gave me water and everything.
it just kind of
And the
came out.
water fixed it.
Oh, no kidding.
Yeah.
Yeah.
I can-
Yeah, I didn't know that could happen. That's kind of scary, huh?
It is.
Yeah. Breathing, not breathing is bad. Remember, that's the takeaway. Cool. So with that in mind, I want to have a little aside here. And I want to talk about artificial ventilation. And specifically, I want to show this short video on how ventilators work, which might not be the way that you assume that they work. So as you know, in recent years, ventilators have been in our minds a little bit more in the forefront than they had been historically because of COVID, because of the need to use them for a lot of people, because of shortages of them in certain cases. Being on a ventilator is, I want to emphasize that it's really traumatic and terrible. And having said that, it's life-saving. Like if you need to be on one, it's good, you want to be on it. But you're unconscious and you have all of these tubes going down through your pharynx and your larynx, et cetera, that really can cause a lot of damage. Again, it doesn't outweigh the fact that it saves your life, right? But it's not like I think sometimes ventilators are portrayed as like this easy fix. Oh, you're having trouble breathing, just get on a ventilator. But it's actually a lot more complicated than that and can have a lot more long-term negative effects. So let's take a look at how this works mechanically. And I need to adjust my volume here. Just bear with me for a second. That works. Let's see. Cool.
In the 16th century, Flemish physician Andreas Vesalius described how a suffocating animal could be kept alive by inserting a tube into its trachea and blowing air to inflate its lungs. In 1555, this procedure didn't warrant much acclaim, but today, Vesalius's treatise is recognized as the first description of mechanical ventilation, a crucial practice in modern medicine. To appreciate the value of ventilation, we need to understand how the respiratory system works. We breathe by contracting our diaphragms, which expands our chest cavities. This allows.
All right. So just some background information as we're talking about the respiratory system. Thoughts, comments, questions on that? All right, let's talk about the trachea next. So the trachea descends superiorly from the larynx and into the mediastinum. Remember, this is the cavity in your thoracic cavity that includes the heart and some of the surrounding tissues associated with the heart. It's kind of the cavity nestled kind of between your lungs superiorly and includes the trachea. As you know, it has these cartilaginous rings that keep it open, and it's lined with pseudostratified ciliated columnar cells, as we mentioned earlier. Let's see here. One important note here, this is kind of wordy, and you can see this better from lab yesterday, but remember these rings are C-shaped such that the back of the trachea does not have these cartilaginous lungs, and that's where the esophagus runs. And so basically, we have this rigid structure, except there is a place in the posterior where there is some flexibility, and that's specifically because your esophagus changes its width dramatically when it's resting or when you're swallowing, and so that just allows a little bit more flexibility between expansion of the esophagus and the trachea. Also, this allows the trachea to not only have some flexibility under those circumstances, but also when you're forcefully expelling air, it allows the trachea to expand a little bit more too than it would if those cartilaginous rings were complete circles. And so, for example, when you're coughing or sneezing, you're trying to get as much air out as you can, and it helps to essentially widen that airway a little bit. Let me see here. There's one note there. I'm just being very judicious about what we're talking about today. I'm going to skip over the microscopic structures of the trachea for now, because we covered this in lab yesterday. So this should look very familiar to you from lab yesterday. You looked at microscope slides that show exactly that. So I'm not going to talk about that now, in favor of talking about the more gross anatomy of this. And so here you can see the trachea descending. There's a special cartilage right here that kind of looks like underwear. That's called the carina, and that is a special piece of cartilage where the trachea bifurcates into the two primary bronchi. And again, you saw this yesterday in lab. Notice those primary bronchi then branch into secondary and then tertiary bronchi. Sometimes these are called the main, lobar, and segmental bronchi, but I usually just use primary, secondary, and tertiary because I think it's a little more intuitive. So basically you have this passageway that keeps splitting. Every time it splits, it gets smaller and smaller and smaller and smaller. And then the smallest bronchi are called bronchioles. So this is an important term there. And they can be very, very small, so like a millimeter in diameter. And these further divide into the terminal bronchioles, which are the smallest of these structures. And they could be even half of a millimeter. So if we look at this another way, we can see how essentially it's just a series of splitting airways. All of these have names. You don't need to know those names, but remember, takeaway message is that in humans, we have two lobes in our left lung, the superior and inferior. In our right lung, we have the superior, middle, and inferior lobe. And you can see how the bifurcating pattern of the bronchi match they essentially split these airways into the respective lobes of the lungs. Bless you. Yeah, I think that's all I need to say about that. For now, I want to get to make sure we have time to talk about the alveoli. So just as a kind of bit of review, in the beginning, I described the passageway of your air is going from your nose to your nasal cavity, pharynx, larynx, trachea, and bronchi. Knowing what you now know, we can expand this, and we can show the pathway of air through the conduction zone in much more detail. So it enters through the nose, then to the nasal cavity. Then we can split the pharynx into the nasopharynx, oropharynx, laryngopharynx. Then we have the larynx and the trachea, and then we know that the bronchi, we start with the primary bronchi, secondary bronchi, tertiary bronchi, then to bronchioles, and then to the terminal bronchioles. So make sure you know all of these structures, generally know the differences between them, and of course know the order through which air passes when air is either entering or exiting the lungs. All of these structures are part of the conduction zone. So not yet have we conducted any gas exchange. Rather, all of the air is being channeled where it needs to go. And in that process, it's being filtered, it's being warmed, and it's being humidified. And that brings us to the respiratory zone. Before we get there, what questions do you have about the conducting zone? All right, let's talk about the respiratory zone. So the structure that we're going to focus on here is called the alveoli. They look like bunches of grapes. And you can see our bronchi get smaller and smaller until we get to these terminal bronchioles. And these terminal bronchioles end in these clusters of alveoli called an alveolar sac. And that is essentially like a bunch of these, what I'll call grape-looking structures at the end of these bronchi. So bronchi are one-way passageways, right? They don't form a loop. Rather, they come and they terminate here in the alveoli. And the alveoli are super important because this is where gas exchange happens. This is where oxygen and CO2 cross from the blood to the air in the alveoli. Because of the structure of the alveoli, because they're all of these little, basically spherical structures and they're clustered together and they fill up our lungs, if you took all of that surface area and stretched it out, we have something like 400 million alveoli, and the surface area would be about 140 square meters, which is approximately the size of a tennis court. So think about that. Every time you're inhaling air, let's say you're inhaling a couple liters of air, that air is spread out across the surface area the size of a tennis court so that gas exchange can happen efficiently. Of course, as you increase surface area, you increase the amount of the efficiency of gas exchange, how quickly gas can be exchanged. So super, super cool to think about this giant surface area, all kind of structured within this small cavity in our chests. Let's see. within these alveoli, there's 3 different cell types I want you to know in the alveoli. So getting down to the microscopic structure. The first is what we call type 1 alveolar cells. And these are these really thin, simple squamous cells. So you saw these in the histology lab at the very beginning of the semester. You saw these in lab yesterday as well. These are the flat cells that make up the surface area of the alveoli. Notice they're simple, so they're not layered. We want a thin layer for gas exchange, and they're squamous, so they're flat. And you can see those cells kind of forming these clusters here. Now notice that, of course, this is a transfect of all of these clusters. You have to picture kind of all of these in 3D getting cut. We also have type 2 alveolar cells that are a special kind of cell that releases something called surfactant. In my physiology class, I actually talk a lot about surfactant. It's a super cool, like basically lubricating viscous liquid that allows your lungs to fill efficiently. And there's a lot more to it than that, but I'll just leave it at that for now. Interestingly, we don't produce surfactant till very late in development, basically right before birth. And so for a long, long time, when premature babies were born, they wouldn't be able to breathe and they would suffer and often very serious consequences, simply because they didn't have surfactant. And one of the most kind of elegant medical breakthroughs of the 20th century was discovering that that was the problem and that you could artificially make surfactant and basically inject it in the preemie baby's lungs, and they would be able to breathe remarkably better. So really simple solution that saved a ton of lives was the invention of artificial surfactant. Finally, the third cell I want you to know in the alveoli are macrophages. So this is a type of immune cell that phagocytizes particles and invading bacteria and things like that that we don't want to get into those air sacs. So as you know, through the conducting zone, your air is clean before it gets to your lungs. However, it's not a perfect process, of course. Stuff does get into your lungs, and we have some immune cells that are hanging out down there, ready to gobble it up and get rid of it. Go ahead, yeah. Say it again. Mm-hmm. Oh, yeah. Yeah, it can happen all along the airway for sure. But I think in that case, I don't know popcorn one specifically, but based on what I do know, I would imagine it's damage to these alveoli. One thing that can happen, a good example kind of going with what you're saying is smoker's cough. People that have smoked habitually for years often just have a kind of constant cough. And that's actually because the cilia and the trachea no longer work. And so you have this mucous membrane in your trachea, the cilia are constantly moving mucus up and then into your oropharynx, so you can then swallow it and digest it so it doesn't go down. Those cilia get damaged and all that mucus basically is just constantly dripping down into their lungs. So they're constantly having to cough to get it up because the cilia aren't working. So there's lots of ways that those, don't smoke, it's bad. And then for vaping specifically, I know it's damaged the alveoli, but I don't know exactly what that looks like. Yeah, and you saw in lab yesterday, we have a cancerous lung tissue and lung with emphysema. And I'm going to actually show you an example of that in just a second. Actually, we need to wind this down. What do I want to talk about here? Give me 1 second. Actually, we're good. I want to show you what this looks like so we have these alveoli where the gas is filling up these little spheres of air, and they're closely associated with these capillary beds where that gas exchange happens. And so without getting into too much of the physiological details, know that these alveolar clusters are surrounded and closely associated with these capillaries. And if we look at this in a microscopic level, there's a lot of information in this slide, but what you can see here is the type 1 cells, these simple squamous cells making up the wall, I'll call it, of the alveolus. Then we have the type 2 cells, which are secreting this surfactant that keeps it lubricated and inflating and deflating efficiently. And then we have a macrophage here, these little guys wandering around trying to pick up stuff that we don't want there. And of course, these are closely associated with these capillaries And that's where the gas exchange happens. We have O2 moving into the red blood and into the blood in the red blood cells, and CO2 moving out. I think that's all I'm going to say about the alveoli. That's a whirlwind tour. Yeah, I've got a little bit more on diseases, but actually, instead of talking about that, I want to show you just a couple video examples here. So I'm going to pause the recording again. So the question is about how you can essentially train your lungs to be more efficient. And absolutely, training exercise can make a huge difference. It can increase the musculature associated with the lungs. It can increase the surface area of the lungs and all of those things. What was the first part of your question?
If everyone can inflate it like a.
Oh, no, and you can, so that actually is an important measure of how healthy your lungs are. Take human physiology and do a whole lab on ventilation and how much air you can, and you can't actually, what's cool is you can't actually exhale all of the air in your lungs. You need to keep some in there. Otherwise, they would just completely collapse. And so it's really important that you actually don't ever suck all of the air out of your lungs. And the amount well, I don't want to get into it, but take human phys. You'll like it. Yeah. Go ahead, Jane.
Lots of smokers' lungs.
Uh-huh.
Is that like someone that died from that problem, or can they still live with that lung?
Good question. Yes, you can up to a point, for sure, but it decreases your capacity to perform gas exchange, right? So you can live with it for a remarkably damaged lung. You can live with one lung. Lots of people live with one lung, perfectly fine, healthy lives. So it's really adaptable, but it reaches a certain point when that, and then often that's combined with other ailments, like it's part of a whole suite of symptoms that goes with it. And that doesn't even get into cancer either. So yeah. Go ahead, Drew.
Is it possible for that to heal over time?
It can improve, but it won't ever like fully get back for sure. And that's, they're showing an extreme example here. So something that bad is never going to get is never going to get functional again, basically. So yeah, emphysema is pretty irreversible, unfortunately. Good questions. All right, well, let's wrap it up. I'm going to give you your quiz back in just a second. So quiz 6 you took yesterday, I've got it graded for you, or excuse me, Monday. Apparently I can't count. The first set of questions had 7 questions. I said it was worth 14 points, but there were only 6. I don't know. Late in the semester, so everyone gets 2 points on the quiz. So you're all starting off with 2 points. The class average was 24, just about 25 out of 30, so 83%. You can see here the mean, and you can see the distribution of the class. Had a handful of perfect scores on this. Keep this handy. This is great review for your final exam as well. Let me know if you have any questions on it. Otherwise, as we wrap things up, do your course evaluations for this and all your courses, please. That's kind of psychedelic. Should have started off class with that. Few reminders. Danny has a tutoring session. What days today? He'll have one tomorrow in Beagley Library. And then he has a special session on Monday in preparation for your lab practical on Monday afternoon. Oh, wait, whoa, slow down, Tiger. I'm going to keep the lab open until 8 p.m. on Monday. So I'm going to do it as late as I can. It takes me quite a long time to set up for the practical, but I wanted to give you as much time as you can. So please plan accordingly. Don't plan on studying late night for the practical. Unfortunately, there's just nothing I can really do about that because I need to get in there at some point and set up for the morning. So Danny will be there on Monday in the afternoon, and it will be open till 8 p.m. on Monday. I'm going to go in there in the evening and get things ready. Remember, the class is taking the practical at 2 different times, half of you at 8:30, half of you at 10. And those times are on your most recent class programs. There is no additional program for Friday, though. So just see the class program for today for that list and all of the previous week and a half or so. Otherwise, I've got a ticket out for you. I'll give you back your quizzes, answer any questions. And yeah, thanks everyone for a fun semester. And as I promised, your last Venn diagram, this is probably one that you're all able to relate to right now in these final weeks of the class.